right, well, thank you all for coming. I know the summer is a busy time for everybody and field season for some, and then some people are graduating, and so yeah. But I really appreciate your taking time out of your busy schedule for this. Glenna completed her undergraduate degree at Western Washington University, and there she had the opportunity to go on a research vessel and study DC ecosystems. And so when she reached out to me, she was really looking to work on DC ecosystems. And I had the unfortunate task of telling her that I didn't really have anything for her in that area. And so we talked a little bit more, and we talked a little bit more about this project that she'll be talking about in the Florida Keys. And she was really interested in jumping on that. And so that was fantastic. The other thing Glenna has is a lot of ideas. She has a lot of ideas. She's very driven, motivated, and I would say that, yeah. And had a really good idea of what she wanted to do when she came into the center, which is working on habitat suitability models, and then using them to either determine like species location or restoration activities or whatever it would be. But she really wanted to learn more about habitat suitability models. And so with the help of Marine Lab, our collaborators, and our other collaborators are Oregon State University, along with her committee members, Tom Lippman, Easton White, and myself, we kind of honed all of her great ideas down to using habitat suitability models for coral restoration. And so with that, I will.
All right, thank you everyone for coming to see the work that I've been doing for the past 2 years. My master's thesis focusing on fine-scale niche modeling, growth and disease cover trends of long-term outplanted staghorn coral in the lower Florida Keys. So to start off with, let's talk about staghorn coral. It's a fast-growing, long-lived coral. What does this mean? It means that it grows 10 to 20 centimeters per year, while most corals grow 5 to 10 centimeters a year. So they grow a lot, and they are long-lived. We know that this coral can live centuries long. The species itself has existed for 6.6 million years. It is a stony reef-building coral. That means that from this coral species, or stony corals, the reef is built on top of them. So without a reef-building coral, there is no reef. They exist throughout the Caribbean and Bahamas and Gulf of Mexico. Here you can see their range, with the pink being their shallower areas and the lighter blue being some of their deeper areas. They exist to be a few meters deep and can go as deep as 73 meters deep, going into the Mesophotic zone. Over the past half century, their population has decreased dramatically, resulting in them being labeled critically endangered and protected under the Endangered Species Act. The reason for their decline can be mainly attributed to bleaching events from marine heat and cold waves, disease such as black band disease, white band disease, and Caribbean cilia infection, as well as brown band disease in the Southern Caribbean, pollution from anthropogenic runoff, as well as corallivores. Corallivores are organisms which eat and chew on corals. Here you can see an example of one is a damselfish. There are also snails and brittle stars which will chew on the coral. This can cause lesions in the tissue which allow for disease to enter. In order to fight against this decline, there's been a suite of restoration efforts put forth to try and restore their populations. The main form of this is coral outplanting, where young corals are grown in nurseries on land and are then put out into the marine environment to help build the reef. Here you can see some coral trees. This is where the young corals are getting introduced to the marine environment and getting them acclimated, and from there they will then later be put onto the reef. There has been a lot of genetic focus towards this to build more resilient corals. There are these clonal colonies, so genetically specific types of staghorn coral called genets. There's been a focus on getting genets that have shown to be more resilient to marine heat waves and cold waves and disease, as well as providing diverse genets so that if disease or any sort of disturbance does happen, it does not wipe out the entire reef. There have been antibiotics developed to fight against bacterial infection and certain physical structures to help discourage corallivores. Lastly, one of the more important things that I want to talk a bit about is how we've been looking at optimizing monitoring techniques. At CCOM-JHC, we've been working at comparing different forms of monitoring, from in situ stereo imagery and scuba diving, to types of echo sounders, multi-beam and single beam, and unmanned aerial systems, or UAS imagery. Here, we have been in the Florida Keys collecting data using these three different forms of data collection. And here you can see some examples of that. And there's certain trade-offs when doing these different types of monitoring when it comes to resolution and detail. So for example, our in situ stereo imagery has very fine detail, but the amount of time it takes for us to survey one site, we can survey through echo sounders and UAS imagery in a matter of minutes. So through this, we're on our way to understanding how's the best way to monitor corals and to understand coral abundance changes in the future. When I joined Jen's lab, I became interested in using this data to answer the question, where should we outplant these corals? This is where we introduce habitat suitability models. Habitat suitability models use environmental and biological surveys in order to project areas of organism occurrence, range expansion, and growth. There have been some habitat suitability models already in development, and they have projected that staghorn corals do better in the lower and upper Florida Keys in areas of the fore reef with moderate flow, and turbid waters have shown to be helpful to mitigate temperature fluctuations and UV radiation. When we were working with Mote Marine Laboratory and I came to them talking about habitat suitability models, they're the ones who are doing all these outplanting, and I wanted to see what would be helpful for them. They said that the habitat suitability models that have been put forth thus far haven't been super helpful. There's a few issues with them. One, these habitat suitability models use a really large resolution data size, so about 1 kilometer or so long. It can give site recommendations that are either super big or far away where it's not feasible and economic to go out and outplant and recurringly monitor those sites. Next, these suitability models use wild coral populations, and we know that outplanted corals and wild corals respond differently to disturbance. Lastly, growth and survivability can vary widely within a single site. Where Mote Marine Laboratory has been outplanting coral, you'll see one that's healthy, growing, no disease, and then a few meters off another staghorn coral completely decimated by disease. And it's those fine scale differences that we need to better understand in order to help build more reef. Lastly, there's been very few long-term studies on coral outplants. A lot of research done on coral outplantation happens in the first year or two of outplantation. This is important to know because corals only start reproducing after 3-ish years, and that is what we're aiming for. We're aiming to build reefs that can reproduce and self-sustain themselves. In the few long-term studies that have been done, we've seen that growth and survivability decrease dramatically after 2 years. So this was something that I really wanted to focus on, was to make sure that when building these models, I would look at older corals. So this all led to the aims of my thesis. One, to look at the coral cover and growth trends in the years following outplantation out into the far long-term outplanted corals. Next, to look at what fine-scale seafloor terrain characteristics could be attributed with the variation in healthy coral cover and growth. Lastly, I wanted to see how average annual high-intensity events influence these cover and growth trends in order to see what we can and cannot account for, from seafloor terrain attributes, which we can account for when outplanting, to high-intensity events that we have no control over. So going into our data collection, as I mentioned, we've been working with Mote Marine Laboratory. They've been outplanting corals throughout the lower Florida Keys. Here you can see where their lab is on Summerlin Key. We surveyed 8 of their outplant sites, 4 of which are in sanctuary preservation areas. S4 and S6 are in the Sand Key Sanctuary Preservation Area, and T3 and T16 are in the Eastern Dry Rock Sanctuary Preservation Area. All sites we surveyed in the summer of 2022, and we additionally surveyed ICC1 in 2021. All sites were in the fore reef and in spur-and-groove terrain. Spur-and-groove terrain refers to this type of terrain where we see these spurs, where the corals are growing on, cut through by these sandy grooves. So that's an aerial image, and here you can see in the water these staghorn corals growing on the spurs, and here is the sandy groove cutting through. So all of our sites were on spur and groove terrain, except for ADAC1, which was in the patch reef. Corals outplanted ranged to be outplanted as far back as 2017, so about 5 years prior to surveying, to 2022, so just a couple of months before we went out to survey. Surveying took a form of photogrammetry. This is where we took, or I should say more specifically, Jen was taking planar images using a Canon DSLR camera. And here you can see the rig with 2 Canon cameras right there. We did a lawnmower survey pattern both laterally and horizontally, resulting in maximum image overlap. We did this in 10 by 30 and 10 by 60 meter plots. After we did that, we took those images and coming back to New Hampshire, we uploaded them into the software Agisoft Metashape. These images are then able to be stitched together to create orthomosaics, which are ortho-rectified photomosaics from an image collection. So here you can see a 10 by 30 meters survey site. And here's a larger example of a 10 by 60 meter survey site. In addition to this, because the images are taken at multiple angles, a point cloud can be made, and then from that you can make a 3D model. So here we're getting more sideways view of Staghorn corals. From these 3D models, you can create digital elevation models. Here you can see the color referring to how deep it is, and you can actually see the Staghorn coral and other coral species influencing that bathymetry. After that was done, I exported the digital elevation models and orthomosaics and imported them into ArcGIS Pro. I resampled the DEM to the coarsest resolution size I was able to get from all of our sites, which was 8 millimeters. And I just want to say that because our finest scale digital elevation model was like 0.2 millimeters, so very, very fine scale. It's also important to mention, as I said, most habitat suitability models are working in the resolution of kilometers or at best meters, so extremely fine scale. After these were imported into ArcGIS Pro, I quantified coral cover by creating these polygons of the healthy and total tissue cover. So here you can see the Staghorn coral. The dark pink is referring to healthy tissue cover, which I was able to see as being dark orange tissue, while the lighter pink is covering the not healthy tissue cover. This is tissue that's been diseased or covered with turf algae. I then extracted from the digital elevation model depth, slope, aspect, roughness, and curvature values from the 10 centimeters surrounding each coral. So here you can see that buffer surrounding each coral. Depth, obvious, just how deep the coral is. Slope referring to the angle at which the bedrock is. Aspect refers to what cardinal direction that slope is facing. So north being 0 degrees, south being 180 degrees. Roughness is referring to how much for each cell, how much the surrounding cells are changing dramatically in value. So a more rough terrain means there's a lot of change, and not rough means that's pretty flat. Curvature refers to how convex or concave that change in roughness is. Other environmental characteristics I wanted to add included distance from other Staghorn corals, how densely packed those corals were to each other. I also wanted to include how far away the Staghorn coral was from the spur-and-groove interface, which I call the reef edge. This reef edge could be about a foot to a few meters of a drop off. So I wanted to see how far away it was from that. And then also distance from coast. Okay. Like I said, I wanted to include high intensity events. This includes marine heat waves, marine cold waves, and high wind events. I took a definition from Hobday et al. On what marine heat waves are being standardized to, which is days of 5 subsequent uninterrupted days where the average temperature for that day exceeds the 90th percentile taken from a 30-year historical baseline record. I applied the same definition to marine cold waves being anything going below the 10th percentile of a 30-year historical period. High wind events I defined by days that the average wind exceeded 12.78 knots, which is the average sustained wind speed from a tropical storm. I was able to collect this data from the Sand Key, Key West, and Sombrero Key NOAA buoys, which you can see right here. So I collected both the period that the corals were outplanted, those temperature and wind data, as well as the 30-year historical baseline from those buoys. I then wanted to avoid collinearity with time, so I had to transform the data. I also wanted to look at how long these heat wave and cold wave events were occurring, as well as how intense. So a 5-day marine heat wave where it's only going a degree over the 90th percentile versus maybe a much shorter period of time where it's going several degrees over the threshold I set. So intensity is the number of degrees exceeding the 90th percentile, going below the 10th percentile, or going above that 12.78 knots. Duration being number of days that event was occurring. And then I divided intensity by duration, and then I divided that by the number of years that a coral had been outplanted. So those are all my covariates that I'm using to predict. And here we have my response variables. Here there is percent healthy cover. This refers to what percentage of a staghorn coral outplant has healthy tissue cover. So 100% means there's no disease, that coral's thriving, it's doing okay. 0% means that coral has been completely destroyed by disease and for all intents and purposes seems to be just dead. I also wanted to calculate total growth as well as healthy growth. Total growth referred to the total change in skeletal size per year since outplantation. So this will include diseased tissue, but I also wanted to look at where corals are continuing to grow despite being infected by disease. So I looked at the change in healthy coral tissue size per year since outplantation. I excluded fragments because despite the fact that coral fragments will become corals in their own right, that's asexual propagation, I would not know when that fragmentation had occurred. So I didn't want to include fragments, and I didn't want to include wild coral populations, which I only saw at one of my sites and was really easy to tell wild from coral outplants. This resulted in a total of 1,168 corals that I used in my model. So going into data analysis and the structure of my model, we got to do a little bit of stats review. I hope you all will enjoy my little lecture. So we have a simple linear regression. We all learned about this in grade school, y equals mx plus b, or in a more advanced term, b naught, which is our slope intercept, or y-intercept, b referring to the slope, and then we have our independent variable, or in habitat suitability models, we call it a covariate, and then our error or residual term. Then we have multiple linear regression where you're having multiple things to project your response variable. So for example, if you wanted to predict how a student might do on an exam, you would include the number of hours they studied and maybe how much sleep they got before the exam. So that's a general linear model. We all know and love it. It has a normal or Gaussian distribution of data, and our response variable is unbound. But the thing is, that doesn't always work for all types of data. In the example that I just used when it comes to an exam, you can't have a student who maybe studies a lot and gets a lot of sleep and therefore gets 250% on the exam. Also, if you had a student who didn't sleep very well and didn't study at all, the student can't get negative 10%, or I hope a teacher would never give negative 10% on an exam. So we can't have the response variable be unbound. So we have to find it. This is where generalized linear models come in. It's an extension of general linear models, and it allows for data that is distributed with a beta distribution, Poisson distribution, or binomial distribution, and it does it through a link function. This allows for our response to be bound, and it doesn't have to be continuous. It could be categorical. Next, we have mixed models. Mixed models have a bunch of different names: mixed effect, nested, random intercepts, etc., but they all essentially mean the same thing. It means that we are allowing either our intercept term, our slope term, or our residual term for that value has its own probability distribution. It could have a range of values. And we do this whenever there's, for whatever reason, we're not entirely sure we can pin down what that value would be. So using my model for an example, we have all those covariates I mentioned: depth, slope, aspect, etc. And I also wanted to include site. There are certain variances between my site that my model wouldn't capture. For example, dissolved gases, nutrients, temperature fluctuations. These all could vary across sites, but I didn't collect that data. So I added random effects per site, and a maximum likelihood estimate was able to estimate the best slope for that covariate site. So this is my model, a generalized linear mixed model. Like I said, my response variable is going to be total growth, healthy growth, and percent healthy cover. For growth, I used a Gaussian or normal distribution, but because percent healthy cover is bound between 0 and 1 or 0% and 100%, I used a beta distribution via a logit link. This helped to keep everything bound so I didn't get 110% healthy cover or negative 10% healthy cover. I also wanted to take a look at long-term versus short-term environmental covariates that could influence my corals. Because so much of the research has been done in the short term, I wanted to see if perchance in the short term corals had certain needs, but then after a few years, maybe they had different needs or certain things become much more important. So I decided that if I saw any radical differences in the short term versus long term, I would run submodels of those age groups. And my model selection took the form of excluding highly correlated variables. So let's get into the results. Looking at percent healthy coral cover, this refers to, as I said, the percentage. Over here is 100% healthy coral cover, and this is 0%. And as you can see, there's a pretty big difference between corals less than 2 years and older than 2 years. For corals less than 2 years, healthy coral cover was around 100% with maybe a few outliers. But after 2 years, there became a dramatic decrease in percent healthy coral cover. We also start to see corals that have 0% healthy tissue cover. So completely covered with disease or turf algae. So I decided, as I mentioned, to run submodels of corals less than 2 years and older than 2 years. And what I saw when I ran a model with less than 2 years is that no covariates were projected to influence healthy coral cover. This is important because it means that the research that we're doing in the short term in a model was not able to project where corals would do good or bad. So we need to start focusing more on older corals in order to understand where corals will do best. For the corals older than 2 years, I did have covariates that were shown to be significant, and I'll get to that in a minute. But looking at my habitat suitability model for all corals, we saw that Staghorn corals had better healthy cover when they were farther away from the coast. There could be a few reasons for this. We've seen in other habitat suitability models for other coral species similar results, and it's been attributed to being farther away from anthropogenic runoff and pollution. The other reason could be that the corals that were farther away from the coast were also corals that were in the sanctuary preservation area and were also farther away from the middle keys, where the middle keys have been shown to be especially bad for Staghorn corals. So those could be some additional reasons for why corals farther away from coast did better. Next, corals that were next to the reef edge were shown to do better. So remember, the reef edge is where there's that spur and groove interface. The reason that this could be is because, as I mentioned, there are these sandy groups where the water is carving through the reef. And so there's a higher water flow and a more higher water flow, bringing more nutrients, cooler water, etc. So it's likely due to being moderate water flow, and Mote Marine Laboratory themselves had observed these changes, and they started preferentially outplanting corals near the reef edge because they saw they were doing better there. Next, looking at depth, corals that were in deeper water did better, excluding the sandy grooves, which are not suitable for corals, but anywhere else, corals did a lot better in deeper waters. And this is likely due to the fact that when you're deeper, there's less temperature fluctuations, it's a bit cooler than when you're up shallow, and there's less UV radiation. UV radiation is its most damaging in the first 6 meters or so of the water column, so deeper than that can be very beneficial for corals. These results were shown to be significant for older corals, so in the long term, this is especially important. Next, roughness. Now, as you can see, roughness is very variable in the finer scale, so zooming in a little bit. Higher roughness was indicative of more corals, sea plumes, other organisms, while less roughness was where there was more sandy terrain. My model showed that less roughness was better for healthy coral cover. There's a few reasons for this. One, if a coral was in terrain with a lot more corals and organisms around it, there was a higher proximity to other organisms that have disease and could thereby be spread through proximity and touching. Next, more complex terrain is the location that corallivores like to inhabit. So if you remember me mentioning this little damselfish, the snails and the brittle stars, they like to be in more complex terrain, make sure that they're nice and covered. So more complex terrain could mean that being around more corallivores that chew on the coral and allow disease to be taken in. Next, when a coral is surrounded by other organisms organisms and water is flowing through, the other organisms can help uptake nutrients and the water is thereby sort of flows around and it cannot flow smoothly to the coral, bringing all those nutrient and gases. So it's essentially a competition from other organisms for those nutrients and gases. Again, these results were significant for older corals, so this is especially important to consider. Next, let's look at growth. As I said, I did total and healthy growth, and unlike percent healthy coral cover, we did not see any sort of dramatic change after 2 years. When I'm going over my model results, I should mention that these covariates that were shown to be significant are significant for both healthy and total growth. I didn't really see many covariates that was significant for one, but not significant for the other. So distance from coast, so being farther away from coast and less rough terrain, similarly were shown to be good for coral growth. Being close to the reef edge was not shown to be significant, but slope was. This is possibly due to the fact that when it comes to healthy coral cover, we're trying to be in places where there's less disease. And so the coral just needs not stagnant water, it just needs a bit of water flow. But slope could be referring to how much water flow is occurring. If it's steeper, that means that there is a deeper channel and there's more water going tightly through a constrained area. Meanwhile, if there's less slope, that means that the water could be more dispersed. So this indicates that corals that are growing better are in more slope, in terrain with the steeper slope and therefore stronger water flow, bringing more nutrients and more dissolved oxygen and important stuff. Curvature, like roughness, is really variable in the fine scale, so zooming in a little bit. Like roughness, we see that the major differences are occurring where there are organisms surrounding versus sandy terrain. Convex environments are where there are more organisms, while concave environments are where there are less organisms around. And concave environments were shown to be better for growth. So like I said, convex environments could be a coral is just being choked out by all these organisms surrounding it, and concave environments mean that the staghorn coral has more space around it. All right, high intensity events did not have an influence on healthy coral cover, but it did have an influence on growth. So let's take a look at that. Model average coefficients refers to that value of the slope. So positive means it has a positive effect, and negative values means it has a negative effect on that. So here we can see high wind events had a positive influence on staghorn coral growth. And this is kind of surprising. There's a lot of research showing that high wind events are bad for staghorn corals. It increases runoff, causes fragmentation where lesions can cause, and it in general has been shown to reduce growth. But high wind events have shown to not always be bad. It increases wave action and water mixing. It also, while causing fragmentation, those fragments become corals in and of themselves. So it increases asexual propagation. Lastly, and possibly most importantly, it induces ocean cooling. So it can stop those marine heat waves, which have shown to be very destructive to corals. Something else to consider is that I used a lower threshold for high wind events than other people did. I did that because my sites were shallower and I wanted to include tropical storms instead of just hurricanes. Other papers use a threshold of 20-ish knots, so going more into the hurricane range of things. So my model could also just be saying that moderately high wind events, so not extremely destructive hurricanes, but tropical storms are helpful for corals. Next, my model results showed that marine heat waves were bad for coral growth. And that's not surprising. We know that due to climate change and the increase in marine heat wave events, this is shown to be especially bad for corals and is driving a lot of their decline. What might be surprising if you look is to see that marine cold waves were shown to have a greater degree of negative effect on the staghorn coral. But it's not surprising if you know a little bit about the history of the Florida reef tract. Back in 2010, a marine cold wave hit the Florida reef tract and greatly decimated the corals throughout them. So it seems that corals are actually more susceptible to marine cold waves, but we haven't had as much research going into that because due to climate change, marine cold wave intensity and duration is likely to decrease. This isn't a thing to be concerned about because as we move forward, we're trying to heat-proof our corals, doing genetic development so that they are more resilient to marine heat waves. But if we do that, we could possibly be lowering their tolerance for marine cold wave events, And while they're likely to decrease in intensity and duration in the future, they're not going away. So we don't want to make our corals more vulnerable to disturbance and wrinkle waves. Looking forward, we're hoping to do some validations with Mote Marine Laboratory on these models, see how accurate they are, and hopefully we'll be working towards finding locations within sites that can be best for coral growth and healthy coral cover, and find the best places for them to be outplanted. So great, we answered the question, where should we outplant? One and done. But as so often happens when you're doing research, you start to ask the question, how accurate is this? How much can we trust these results? So when I first went a year ago and I was first starting this project, I wanted to make sure that the coral measurements were going to be accurate. When I got in the water, I was measuring the maximum skeletal length of these corals, and then I measured them in the orthomosaics to see how accurate they were. And as you can see, pretty accurate. I also wanted to measure height, and that became a little more variable. Here you can see an example of a 3D model, and here's our Staghorn coral. Very nice, very clean, very clear structure, and bright orange tissue. Here's an example of another model, and while you can still kind of see it, it's getting a little blurrier, it's not as bright orange coral tissue, it's becoming a little unclear. And then here we have another Staghorn coral, which you can barely see. So I wasn't able to include height in my model because I wasn't really able to tell the difference between corals and the bedrock. There also is another issue that I ran into for my models. Like I said, I used depth, slope, and then aspect was broken into northernness and easternness to deal with the finicky circular statistics, curvature, and roughness. But terrain attributes are computationally dependent. There's a bunch of different ways to calculate these values. You have your max, min, mean. In ArcGIS Pro, these are just 6 ways to calculate curvature. There's a bunch of other ways that you can calculate it in other softwares. And surface complexity or roughness has a bunch of different ways you can calculate it. Now, while there are papers giving suggestions for the best ways to make these calculations, these values and these best metrics are usually done on the kilometer scale resolution. I'm working on a resolution that's much finer. We also know terrain attributes are resolution dependent, especially curvature. So a value of a cell here could be very different of the cell here. So which one's the best? Also, terrain attributes are cell calculation window dependent. You can calculate the value of a cell from the surrounding cells, so just one cell surrounding it, a 3x3 cell window, 2 cells surrounding it, a 5x5 cell window. You can go as far up to an 11x11 or 15x15 cell window. 3x3 cell windows amplify those smaller changes, while 15x15 cell and 11x11 cell calculation windows can dampen those small scale changes. So which one do we trust? Next, terrain attributes are platform dependent. In this project, I was using in situ stereo imagery, but we've been also collecting other forms of bathymetry. Which one do we know is the best and the most accurate? And is it transferable? Can my habitat suitability models that I used through stereo imagery also be applied to UAS imagery and multi-beam imagery? We don't know. So it begs the question, how do habitat suitability model results change across resolution, cell calculation windows, and platforms? I haven't answered this question yet, but I did do a preliminary analysis / I messed up a little bit in the beginning. When I was first doing this project, I was using the tools I saw other people had been using in their papers. These tools in ArcGIS Pro, surface information tool, curvature and aspect tool, fit a plane to the cells in order to make those calculations. But something wasn't quite right. I was getting some really extreme values. For example, my slope value, the lowest value I got was 19 degrees, which I know isn't true. I had corals on flat bedrock, so there should have been a fair amount of 0 degrees. When I did a little bit more research, I saw Esri release their new surface parameter tool in February 2021. That's why I hadn't seen it in any of the research I had been looking at. This tool is better than the other ones because instead of fitting a plane to the cells, it fits a surface to a cell. And it also uses larger cell window calculation. And this can cause less extreme values, especially at the finer scale. And since I was working at such a fine scale, I knew that this was the better tool to use. So taking a look at the difference in terrain attributes, Here's fitting a surface to a cell, the new tool I used, and then fitting a plane to the cell, the old two tools I used. And as you can see, slope, the values were near 90 degrees, which I knew the corals were not on. And like I said, the lowest value was around 19 degrees. So I knew this wasn't correct. Meanwhile, when I was fitting surface to cells, I got very different terrain attributes and got values closer to 0. For curvature, the median values were still around the same, but when fitting a plane to cells, I got a lot of extreme values that didn't make sense. When it came to aspect, they were a lot closer to each other. So the same similar distribution of northernness and easternness with the two different tools. Next, let's take a look at model results. When fitting a plane to the cells instead of a surface, curvature was shown to have an influence on healthy coral cover. So that's one difference. And when it comes to growth, I had a few things that were different. When fitting a plane to a cell, distance from coast, roughness, and high wind events became significant. So not only do we get very different terrain attributes, but we also get very different model results. So there has to be a lot more testing in order to see what is most accurate. So moving forward, validation, validation, validation. We want to see what's best in order to collect data. And also we're trying to look at what is best for restoration groups. This past couple of weeks when I went down to the Florida Keys, I also want to see how accurate these terrain attributes are to in situ measurements. So I was measuring depth, slope, and northernness and easternness when I was going down there. And like I said, we're trying to find ways that when I come up with these habitat suitability model results, I can give helpful recommendations to these restoration groups on where to outplant. So with that, I would like to thank my funders at NOAA CCOM-JHC, University of New Hampshire for hosting me, and our collaborators at Boat Marine Laboratory and Oregon State University. And with that, I'll take any questions.
Sorry, we'll take questions for the better audience.
Yes, Liz?
Did you measure flow at all?
So really wanted to. First wanted to try and make something called quad cards, which are made from plaster of Paris. They're not the most accurate way of measuring flow, but it's a pretty cheap way of doing it. They are, like I said, plaster of Paris molds, and what happens is you put them in a location, usually multiple, and you have water flow, which will dissolve the quad cards, and it's a good way of comparing water flow. So we wanted to put some near the reef edge and farther away from the reef bed. We had a few issues come up. One, they fell apart, which probably was the most important, but also when it comes to something like that, you have to make sure that everything is the same. So we would have wanted to have all of the quad cards put at our sites and held over the same days. So if I had quad cards at one site for 5 days and there was a bunch of water flow, and quad cards at another site with very little water flow that day, it would not be a very accurate comparison. So you want them out during the same period of time, which was not feasible. This last time we went out a week ago, we put out tilt meters for 24 hours near the reef edge and further away from the reef edge. So it's going to be a lot more accurate, and I am hoping to do some analysis as soon as I'm done with this. Essentially, that's what I'll be working on this week. So no, I did not measure water flow. We are hoping to do that though.
How many films do you put out?
We had 2 from Frederickson. We put 1 near the reef edge and 1 farther away. We deployed them, collected our data, went to bed, woke up the next day, collected them, and then went to the next site. So 2 per site for 24 hours.
There at one of our sites, there is a master's student who's employed some teachers and they've been left out for 6 months at that site, and they're along the reef and everything.
Yeah.
So we're hoping that we can
Yeah. Is that Florida State University, I believe? Yeah, they were also doing quad cards and tilt meters and a bunch of different forms of collecting water flow. So it'll be a really interesting way to do some comparison.
Thank you. Sorry to.
No, you're good. Yeah.
I had a question, slide 33. When you were talking about your model, one of your variables was site. Is this a categorical variable? How does that work?
So it's categorical and essentially in R, you have to convert it to numerical values, but yeah, you can include categorical covariance as a predictive variable.
So that would be my next question. How would you generalize this model to a different site that wasn't part of your original categories? Would that be possible?
Sorry, say that again?
So your site is just it's a categorical number for one of the sites where you did the work on. But if you were to generalize this model to another site, how would you deal with this variable? How would this would this be possible? Would you need to do another modeling for that site specifically?
Yeah. So when it comes to site, it's essentially just accounting. Okay, trying to get a bit more into mixed model statistics. It essentially is saying that even though two sites might have very different temperatures, it's not going to say that one site is doing so much better, so all of our data of good corals are coming from here. It kind of weights good corals from one site and good corals from another site a bit more equally. So that's kind of why that was used. It's not as used as much when it comes to making projection, like giving recommendations. In terms of sort of the other thing you seem to be asking about applying this model to other things, these models were very much used in the finer scale. So I'm looking at a site, a 10 by 30 or 10 by 60 meter plot, because that is what we're working with. If Moat Marine Laboratory were to say, or any other restoration group were to say, we want to outplant in the Caribbean, can we use your model results on our own? I would say not really, because there's very different things influencing the corals and what my models are being used for. So when it comes to my model results, we're very much looking at the lower Florida Keys at those finer scale resolutions, and the random effects per site is just it's sort of an accounting for variability thing. I'm not sure if that helps.
Without that variable, do you think you still have good results? How important was that variable?
I would say it's pretty important because some of our sites had a bunch of corals that were not doing great, but so let's say we have one site where all the corals are doing great, but it's shallower. And we have another site where not all of the corals are doing great for other reasons, but it's a little bit deeper. It can weigh the deeper corals at both of the sites according to that site. Does that make sense?
Okay, now I understand. It's not categorical in the sense of what site it is. It's category based on how healthy the site is or how.
Yeah, it's also been called nested models. It's kind of like nesting the variables by site.
Thank you.
He asked me a statistics question. I was like, oh boy.
That's what I know.
Oh boy, okay. Yeah.
Because you opened the door.
Oh,
When I look at your slide there, you had said something about the data being normal, and for certain types. And there you've got that your beta I, beta 1 is normally distributed, which is an assumption of regression, but data being normally distributed is not an assumption of regression. And if you look at your graph, I didn't see normally distributed data. I saw uniformly distributed data. So what I'm curious about is you fit admirably different models based on data distributions or whatever, what you assumed. But I think you mean you're fitting them on the betas, the slope, rather than the data. What I'm really curious about is, did you use any sensitivity testing? Because the data were not normally distributed, I'm curious if you used any sensitivity testing to see, did it make a difference whether you or not you assumed a beta distribution or a normal distribution or a uniform distribution?
So you mean like using different logit links functions?
Yeah. Go to your graph, and I think
Of the growth?
Yeah, the example that you gave, I appreciated your stats lesson. No, not this one. Go back. You have a scatter plot that has bands on it. There you go.
Oh, yes.
Okay.
So this isn't my data. This is just a figure I grabbed from. Yeah, so the normal distribution is just how much of the data you have. You know this, but the distribution of the data. So for example, my growth generally had an average per site, was normally distributed around that average. So it was normally distributed in that way, while beta distribution for my percent healthy cover was not normally distributed.
Yeah, okay, so the data that I'm looking at, I accept that that's just an example. If I look along the X direction, I were to prepare a frequency histogram along the X, it's a uniform distribution. If I do the same thing on the Y and do a frequency histogram, it's still a uniform distribution. A Gaussian distribution will have a concentration in the center. And so when I look at that and I say that sort of example, I don't see a normal distribution, I see a uniform distribution. Your data may have been distributed differently, and it's really, really good to be protected against the potential violation of assumptions of regression. But it's still, for me, it would have been useful to say, did you do any sensitivity testing on whether or not a beta model, a beta assumption model fit better than a normal? Did you look at that at all?
No, I have not done. I have wanted to do some autocorrelation spatial data with residuals, which I'm not sure if that relates. That's sort of the sensitivity testing that I am familiar with when it comes to habitat suitability models. And that was something I didn't get around to, but it is something I have been wanting to do. So no, not yet.
Okay.
Yes. Oh.
I can read off of it.
Yes, please.
Jordan Pierce says, "Hi Glenna, great presentation. How did annotate segment the coral used in your study?"
Yeah. Okay. So
We didn't use your segmentation process, Jordan.
yeah, so how did I get the values for the maximum skeletal length or for coral cover? Because
I'm not sure.
okay, we'll just say for coral cover, when you make these polygons, they give you the area of that coral in whatever unit you decide you want to use. So that was the way I was able to do it in ArcGIS Pro. When it came to doing some validation measurements of maximum skeletal length, I just took one of those measuring tape things in ArcGIS Pro and I just measured the max skeletal length. And then each coral was associated with a tile, and that's how I was able to make sure I was being consistent. So when I was in the water, saw a coral, I measured it and I saw its next tile. Tiles are these little white squares right here with a number on it. So I said tile 99 has a coral that's 27 centimeters long. I went into the orthomosaics, found tile 99, and then I measured the coral, and that's how I was able to do a comparison.
Hey, Glenna, sorry, I just got unmuted. Thank you, Chris. Yeah, I was just genuinely curious. So when you're showing these images that are in the current slide, you have them sort of segmented in pink. And I guess my original question was, yes, what tool did you use to annotate, which you said was ArcGIS, and also the data modality. So you mentioned that you were able to segment them and sort of label them using the orthomosaic. So I'm guessing you're using the DEM as a reference for
Jordan,
yeah, we can't hear you.
Oh,
Sorry about that.
there. Can you hear me now?
Oh,
Yeah.
Okay, cool. So ArcGIS was used as a way of labeling the orthomosaics. So I guess my question was the data modality that you used to collect the sort of structural information data. So it sounds like you used the DEM as a source of elevation.
Yes.
Okay. And because you use the DEM, do you think that your structural complexity metrics that you calculated from it would differ significantly, or how do you think they would differ, I guess, if you were to use the mesh or the point cloud as opposed to the DEM?
Ooh, let me think that. So instead of using you might be more familiar with this. The mesh is built from the point cloud, and the DEM is derived from that, is what I'm familiar with. I'll be honest, Jen built the DEMs and then I clicked export. So you're saying a mesh derived DEM versus a point cloud DEM?
Well, so I was just curious, because when you create a DEM, it's sort of like a height fill map. So it's 2.5D. So you don't have the full 3D sort of reconstructed model.
Yeah.
And if your calculations for structural complexity are calculated using the DEM, just curious to see if, in your analysis, do you think it would be worth investigating using the mesh or the point cloud for calculating structure instead of just the DEM?
Yeah, I mean, when I was talking with the data visualization lab, that was not exactly that, but certain questions referring to the differences in DEM development through Metashape and the different ways you could build it was a question that I had never considered. And that is stuff that I am kind of curious about. There was a lot of variability in our model development. So I am interested in doing some comparisons. As for how I would say it would be different, I cannot say I have much of a guess since I was not involved as much when it came to the 3D model development.
Yeah, no worries. That's also a question that a lot of other researchers are really interested in, is the difference between the 2.5D like DEM elevation versus like a point cloud or mesh, just because you do have a slightly different perspective and that it's closer to being true 3D. Okay, cool.
Jordan, we've been working with the data visualization lab, and we created a program. They created a program that I've been testing, where we're using the mesh, the full 3D mesh within that program. And so part of our analysis is rugosity, vector dispersion, fractal dimension, a lot of it's surface complexity. And we found certainly that the 3D mesh is that having a full 3D model is much better than having the 2.5D.
Okay, nice. Yeah, I'm excited to see that. I saw short little snippets online. That's really interesting. And Glenna, final question is with regards to annotating, since you sort of boots on the ground and doing the manual clicking, how long did it take to annotate, your coral species? And do you have any recommendations for, other methods that you might have saw that might help with scaling? Because annotating data is a very time-consuming process.
Yeah, it took a while. A lot of days of just straight clicking around a coral. We have a few different things. We have someone at Moat Marine Laboratory who is also creating 3D models and doing photogrammetry. And they were using a different software where I haven't used it, so I can't say. They essentially said it would they would just circle the coral and then it would just auto tighten the data points around the coral species. So it was a much faster way of getting coral cover data. I have not done that, but it sounds like it would be much faster. So I'm curious about that. What we are hoping to do moving forward is that there have been some works on creating neural networks and deep learning that will be able to identify the coral species for us and get that coral cover data, which if we could run for us would make things far quicker and far more efficient. So I'm hoping to eventually try and see if I can work on doing something like that. But of course, with something like that, you need a training data set. So I'm hoping that my work will not go to waste and hopefully can be used for training some sort of deep learning software.
Cool. Well, thank you for answering my question. I appreciate it.
Of course. Any other questions, Chris? No?
Should I follow up with that one? How many corals did you annotate again? Was it over 1,000?
So the corals that were in my model were over 1,000. So before I knew about it, I had ADAC1 as one of my sites, or ACER. I went into it, and by that point, I was really good at finding corals. So I saw in the middle of ACER these really big, healthy corals, and I just immediately started doing that. And then I later realized that those were not the coral outplants, those were wild coral populations. And the coral outplants that they had done had only happened, I think, a few months before we got there, so they were extremely tiny. So then I later found those ones. So I think those might add a few hundred fragments, probably a few hundred. So we're talking maybe 1,500 corals.
So you're looking at 6 months straight, 8 hours a day?
Yes, yes, we are. I popped in an audio. I popped in an audiobook and I just went to work every day. Point and clicking, point and clicking.
Yeah, well, we're working on creating an annotated data set.
I'm hoping.
The resolution and color and all that make this possible, right? The high resolution DEMs that you make.
Well, the high resolution orthomosaics.
Or
Yeah.
the-
Yeah, I got it from the orthomosaic.
Or the orthomosaics, yeah, sorry. Really make that.
Yeah, I mean, there were a few times, so when you're getting orthomosaics that fine scale, you can imagine the data, the files are large. So when exporting the orthomosaics, they probably could have even be finer scale, but at a certain point, you know, I don't need a gajillion size gigabyte orthomosaic. So I did have to pare down the resolution in order to make the file sizes smaller. And that made it extremely clear. And so, yeah, there was a sweet spot where I had to make sure it was fine enough resolution so that I could identify the corals. Because there were also other corals that looked a lot like Staghorn corals. So I had to make sure it was fine enough scale in order to see the Staghorn versus fire corals. They look a lot like Staghorn if you don't know what it looks like. Any other questions?
Thank you for everyone on Zoom. Thank you for all your questions. And the committee, we have this room for another hour.
Hour and a half, yeah.
For the room for another hour, that the committee can stay behind. And that would be great.
Help yourself to some cookies, please.
Yeah, please take pictures. Yeah. Thanks, everyone.